So without wasting time, now we are going to discuss some of the important things. The very first one is, what is the resting membrane potential? Resting membrane potential is the potential at which the cell is at rest, right? That is called the resting membrane potential. And the next thing, threshold potential and spike potential. I will discuss these two after explaining this, fine? I will tell you the definitions. So for example, this is a skeletal cell. When we touch a person, what happens? A lot of sodium comes inside the skeletal cell, right? As the lot of sodium comes inside the cell, so the resting membrane potential of that cell is disturbed. For example, if it is minus 90 millivolt, then it, for example, for a short period of time, it fluctuates. This fluctuation is called as the stimulant artifact, right? After that, for 1 millisecond, the latent potential generates, right? Then after the latent potential, what happens? These if it is minus 90 millivolt, right, the resting membrane potential, it is disturbed and it goes to minus 50 millivolt, right, which is called the threshold potential. This minus 50 millivolt. Okay, fine. When it reaches the minus 50 millivolt, means threshold potential, then suddenly the sodium gated channels, sodium voltage gated channels open, and a lot of sodium comes from the outside to inside the cell, right? And after that, due to the sodium voltage gated channels, due to a lot of sodium inside the cell, the electronegativity of the cell start decreasing, right? And it reaches up to plus 65 millivolt, right? When it reaches to plus 65 millivolt, then the potassium voltage gated channels open and a lot of potassium go outside the cell from inside the cell, as the cell is full of the potassium, right? So when it goes outside the cell, then again the cell starts repolarizing, fine, repolarizing, and it reaches to resting membrane potential. When it reaches to resting membrane potential, means minus 90 millivolt, again it will not stop, it will go down, and it reaches to equilibrium potential of potassium, right? And this process is called as hyper repolarization. Then for a short period of time, this hyper repolarization occurs, and then again the cell becomes stable, okay, to it reaches to minus 90 millivolt, the resting membrane potential, right? So this is all about the skeletal cell. Now what is the very first thing that I told you? What is the threshold potential? The threshold potential is the potential in which the sodium voltage gated channels open and a lot of sodium come from outside to inside the cell, right? This is called the threshold potential. And the next thing is, what is the spike?
Assalamu alaikum. Today I will talk about why heart is working 24 hours and 7 days. Heart is working 24 hours and 7 days because of pacemaker activity of SA node. The rate of production of impulse is more in SA node than other parts. It is 70 to 80 minutes. The fatigability of heart doesn't occur because of long refractory period in which muscle does not show any response to stimulate. The total refractory period of her heart is 0.53 seconds.
What is calcium calmodium complex? Calcium calmodium complex जो होता है stimulation of the ATP's activity and the smooth muscles जो होती है वो different होती है skeletal muscles में. And the smooth muscle, the myosin has to be phosphorylated for the activities of the myosin ATPase. Phosphorylation of the myosin occurs. Phosphorylation of the myosin occurs has the following 3 points. The first of all, calcium which enters in the sarcoplasm from the extracellular fluid combines with the protein called the calmodium, and it makes the calcium calmodium complex. The second point of the phosphorylation of the myosin occurs at activities calmodium dependent the myosin light chain kinase, and the third and the last point of the phosphorylation of the myosin occurs enzymes. Enzymes in turn the causes of the phosphorylation of the myosin followed by the activation of the myosin ATPase. The fourth point of the phosphorylation of the myosin occurs is the leakage bridge mechanism. Phosphorylated myosin attached to the actin molecule for the longer periods. It is responsible for the sustained contraction of the muscles with the expenditure of the little energy. Relaxation of the muscles occurs due to the dissociation of the calcium calmodium complex. The second topic of the neuromuscular junctions and the smooth muscles. Neuromuscular junctions and the smooth muscle. The first of all is the diagram. Calcium plus the calmodium makes the calcium calmodium complex and it's into the activation of the myosin light chain kinase to the phosphorylation of the myosins and enters into the activation of the myosins. It pays to the muscular contraction. This diagram is the molecular basis of the smooth muscles contractions. The points of the neurotransmitter junctions is that neuromuscular junctions are absent in the smooth muscles. The nerve fibers or axons do not end in the form of the end plate. Instead, nerve fibers end on the smooth muscles fibers in the three ways. The first point is that nerve fibers diffuse on the sheath of the smooth muscles fibers without making any direct contact with the muscles. Diffused nerve fibers from the diffuse junctions which contain the neurotransmitters are released in the matrix which coats the smooth fibers. From here, neurotransmitters enters into the muscle fibers. The second point is that in the some smooth muscle fibers, the axon terminal ends in the form of the many varicosities. The varicosities has the vessels which contain the neurotransmitters. The neurotransmitter is released from the varicosities through the wall in the muscle fibers. The third point is that in the some multi-unit smooth muscle fibers, a gap present between the varicosities and the membrane of the synaptic cleft. The width of the multi-unit smooth muscle fiber gap is 30 to 40 nanometer. This gap is called the contact junction and its function as the neuromuscular junction of the skeletals. Thank you.
Refractory period is a period during which the muscle does not show any response to the stimulus. It is of two types: absolute refractory period and relative refractory period. Absolute refractory period is a period during which the muscle does not show any response to the stimulus, whatever the strength of stimulus may be. It happens because it happens during the depolarization reactions, and so that the second depolarization does not occur. And second is a relative refractory period. Relative refractory period is a period during which the muscle shows response to the stimulus if the strength is increased to a maximum. It occurs during the repolarization state, and refractory period in skeletal muscles. Refractory period in a skeletal muscles is short. Absolute refractory period during occurs extend throughout the first latent period, which is about 0.005 second, and relative refractory period occurs during the second half of the latent period, which is 0.05 second. So the refractory period is 0.01 second. Refractory period in cardiac muscles is a long refractory period. Absolute refractory period extend during the contraction period of the cardiac muscle, which is about 0.27 second, and relative refractory period occurs during the first half of the relaxation period, which is 0.26 second. So the overall refractory period is 0.53 second. The long refractory period of cardiac muscle has three advantages: summation does not occur, fatigue does not occur, and tetanus does not occur.
Assalamu alaikum. Today we will talk about skeletal and smooth muscles. Smooth muscles is present throughout the body, where it serves a variety of functions. It is in the stomach and intestines, where it helps with the digestion and nutrient collection. Smooth muscles derives from both mesoderm and neural crest cells. This is because smooth muscles contributes to many different tissues throughout the body. Organ system involved. Smooth muscles is present in all of the organ system below: gastroenteric tract, cardiovascular, blood vessels and lymphatic vessels, renal and urinary bladder, genital, uterus, both male and female, reproductive tract, respiratory tract, and integument, belly of the skin.
Assalamu alaikum. My name is Taimur Ali. I am the student of OSH International Medical University. Today I will tell you something about SA node. So without wasting any kind of time, let's start. SA node. Due to SA node, heart contacts 24/7 and the resting membrane potential of SA node use 60 milliwatt. When the sodium enters into the cell, the resting membrane potential reaches threshold potential and calcium voltage. Get channels open and depolarize the cell. After sometimes calcium get channels closed and potassium get channels open. Now the cells start depolarizing again and reach the resting membrane potential, and again same mechanism is followed. So depolarization and repolarization in are collectively known as action potential. The potential between resting membrane and potential and threshold potential is called pacemaker potential. The same characteristics are present in AV node and Purkinje cell. If SA node is damaged, then AV node becomes functional. But if AV node is damaged, then Purkinje cell becomes functional. Thank you. Allah Hafiz.
Assalamu alaikum. My name is Shahroz Sadiq. Today's my topic is contraction of muscle and the events occurred during this process. This hypothesis was introduced by H. Huxley and A. F. Huxley and their colleagues in 1954. According to this theory, the thick filament slide past the thin filament slide past the thick ones, so the actin and myosin filament collapse to the greater degree. In this contraction, I band becomes shorter, Z line become close to each other, and H zone disappear. In the process of contraction, the thick filaments attach towards the actin filament. Cross bridges of thick filament pull towards the actin filaments towards the center of salt.
Assalamualaikum, here is Zainul Abidin. I am the student of OSH International Medical University. Today is my topic electrical gradients of different substones in ICF and ECF. Let's start. 3 sodium ion from the cell get attached to the receptor side of the sodium ion, the inner surface of the carrier protein. 2 potassium ion outside of the cell ion to the receptors of the potassium ion located to the outer surface of the carrier protein. Binding of sodium and potassium to the carrier protein to activate enzyme ADPase, and ADPase causes the breakdown of ATP into adenosine diphosphate. ADP release the high energy phosphate, and no energy liberated causes some sort of the conformational change in the molecule of carrier protein, because this outer surface of molecule potassium ion and inner side of the cell and inner surface of the protein molecule with sodium ions faces of the outer side of the cell in association and release the ion take place, so that sodium ion are released outside the cell. ECF and potassium ion are released inside the cell ICF and accept.
Assalamu alaikum. My name is Talha. Today is my topic about actin myosin complex, its activation and deactivation. First of all, when cell membrane or sarcolemma depolarized, then calcium channel open, then calcium from extracellular fluid comes into the intracellular fluid. Calcium increases in the intracellular fluid, then it goes to transfer tubules, then from transfer tubule it goes to sarcoplasmic reticulum, where it opens a calcium pump. From a calcium pump, calcium intake into the sarcoplasmic reticulum, then it binds to the protein called calciquestrin protein. Then calcium went to the transfer tubule from terminal cisternae, then it goes calcium, then enter in between I band and A band, where it binds with the myosin head, and this is a Z line, this is M line, this is a also Z line, this is a light chain, this is a heavy chain. Light chain is also called actin, and it is present at I band, and heavy chain is also called myosin chain. It has H zone. Then a calcium attached to troponin C, which activates the troponin T, which stimulate tropomyosin. Tropomyosin cover the myosin head, but when calcium enter into I and A band, then tropomyosin naked the myosin head, and calcium attached to the myosin. Then calcium attach with the myosin head, then cross bridge develop, tension develop, contraction occurs, then Z line come close together, and H zone disappear, I band decreases in size, A band don't change. This is the contraction, and then relaxation occur when calcium decreases intracellularly. Calcium reaccumulated in sarcoplasmic reticulum, and then calcium release from troponin C. Tropomyosin again blocks the myosin head, and then again contraction and relaxation occurs in this way.
Bismillahirrahmanirrahim. Today my topic is plateau and its significance and the action potential. Let's start. If we discuss about the plateau, what is plateau? Plateau is that stage in which our breathing will become rapid and muscle are cut plus 2 times. If we discuss about the phases of action potential, there are 4 phases of the action potential. First one is the initial depolarization, second initial depolarization, third is plateau or the final depolarization, and fourth is the final repolarization. If we discuss about the initial depolarization, it is very rapid and it lasts about 0.002 seconds. If we discuss about its amplitude is plus 20 millivolt. And second phase is the initial depolarization, which starts after the initial depolarization. It is also very short and rapid. It's also lasts about 0.002 seconds. And third phase is the plateau or the final depolarization. Afterward muscle fibers remain in depolarized state before the further repolarization. It forms plateau or stable period in the curve. It lasts about 200 milliseconds in the arterial muscle fiber, and it lasts about 300 milliseconds in the ventricular muscle fiber. Due to the long plateau in the action potential, its contraction time is also longer by 5 to 15 times than the cardiac muscle fiber. Plateau on the ionic basis of the action potential. Plateau is due to the slow opening of the calcium channels. These calcium channels kept open for a long time through which influx of the calcium take place. Already sodium channel are open through which the influx of the sodium take place. Due to this continuous entry of the sodium and potassium ion into the muscle fibers, producing a prolonged depolarized in the skeletal muscle fibers. The entry of the calcium ion in the skeletal muscle fibers play an important role in the contractile process. That's all. Thank you.
About all or none principle. All or none principle states that the strength of response of nerve cell or muscle fiber to a stimulus does not depend upon the strength of stimulus. If a stimulus is above a certain threshold level, the nerve or muscle fiber will fire. More specifically, there will either be a full response or no response at all. There is no partial response. For proper propagation of impulse to occur, the ratio of strength of stimulus to threshold level must be greater than 1. This is called a safety factor for the propagation. Action potential is always a full response. There is no such thing as weak or stronger action potential. Instead, it is all or nothing process. This minimizes the possibility that information will be lost along the way. This principle was first applied to the muscles of heart, but it was later found that the other muscle fibers and nerve cells also respond to stimulus according to this principle.